Hi there, my name is Alex Wong. I'm an assistant professor in the Department of Biology at Carleton University. My lab works on the evolution and genetics of antimicrobial resistance in bacteria. We mainly work on Escherichia coli. We do some work on other microbes as well, such as Pseudomonas aeruginosa. Both of these microbes are opportunistic pathogens, so that means that they are present in our environments and often don't usually make us sick, but they can make you sick either if you're immune compromised or if you get in touch with the wrong strain. So for example, in the case of E. coli lives in all of our guts, lives in the guts of all vertebrates, but it can make you sick in various ways. It can cause gastroenteritis if you get the wrong strain, and it's also responsible for urinary tract infections, blood infections, and this sort of thing. Pseudomonas is found in all sorts of wet environments. You almost certainly have Pseudomonas in your kitchen sink and in your bathtub. And again, usually it doesn't make you sick. It becomes a problem for people with cystic fibrosis. So people with CF often have a long-standing infection of Pseudomonas, and they'll carry the same clone with them for decades, and it's often eventually the cause of death in people with CF. Pseudomonas is also an important pathogen in hospitals, where it's responsible for things like catheter infections and other infections in immunocompromised patients. In both of these species, and in a wide range of other bacterial species, antimicrobial resistance is a serious and growing problem. So many of the strains that you might encounter, particularly in hospitals, will be resistant to at least one antibiotic, if not more. And when you get infected with a resistant strain, that can make that infection harder to treat. So you may go on a course of antibiotics, that course of antibiotics doesn't work, and so you have to go on another antibiotic. So it means it takes longer to treat, it may mean you're in the hospital for longer, it costs more money to treat you, and there's a higher risk of death. And so overall, antibiotic resistance is a growing problem. It's really most serious in the case of infections like tuberculosis that circulate that are resistant to most or all of the frontline antibiotics used to treat them. And so we're getting close to the point of having strains of TB where you can no longer treat them with antibiotics, which is a really frightening prospect given the toll that TB can take on individuals and on populations. So we're interested in knowing how antibiotic resistance evolves and knowing what the genetic changes leading to resistance are, with an eye towards trying to mitigate resistance either through public health strategies or through the development of new pharmaceuticals. So to me, One Health means looking at health issues in an integrated context. So rather than just looking at it from a purely medical point of view, looking at health issues in a social context and as well as a health context and sort of the much broader social and economic context in which health issues occur. So my lab works a lot on antimicrobial resistance in bacteria. So this is when bacteria that cause disease become resistant to one or more of the antibiotics that we usually use to treat them. And this is a problem because it makes it harder to treat people. Some diseases are becoming very hard to treat at all. So things like tuberculosis, there are strains of tuberculosis that are circulating that we really can't treat anymore because we've run out of antibiotics. So we're interested in the genetic basis of resistance and learning how resistance evolves, but to try to counteract antimicrobial resistance, we really need input from across a bunch of different disciplines. So we need input from public health, for example, in determining when and how antibiotics are used. We have to bring in the agricultural sector because a lot of antibiotics get used in farm animals for growth promotion purposes rather than for treating disease. We also have to include engineering, for example, so things like wastewater treatment and freshwater treatment, the design of surfaces and buildings can all play a role in how antibiotic resistance is disseminated, with an eye towards trying to mitigate resistance either through public health strategies or through the development of new pharmaceuticals. I'll talk about a couple of different projects that we're working on. So one of the main projects that we work on is related to the costs of resistance. So here the idea is that a mutation that that grants antibiotic resistance may also come with a cost. So a resistant strain might not grow very well in the absence of antibiotic, or maybe it doesn't get transmitted between individuals in the present, or rather it doesn't get transmitted between individuals if there's no antibiotic around. So here you've got a strain that's good in one environment, it's resistant to an antibiotic, but it might be bad in another environment, it grows poorly in the absence of antibiotic. And if that's the case, then this might suggest a strategy, a public health strategy, for trying to reduce the prevalence of resistance. So here, if you have growing prevalence of some resistance, you might stop using a particular antibiotic. And if that resistance is costly, then you should expect that prevalence to go down. So we investigate the extent to which resistance is actually costly, as well as to which the extent to which that cost is environment specific. So you might imagine, for example, that some resistance mutation, let's say quinolone resistance mutation, is costly, let's say, on hospital services and in a human body, so in a blood infection, let's say, but maybe that same resistance mutation is not costly in some other environments. So maybe those strains can survive just fine in wastewater or in soil. And if that's the case, you might expect to see resistance persisting in some of these environments, wastewater, soil, even if it is costly in other environments. So that might explain some of the persistence of resistance mutations that we know occurs. The final tool that we have, that we use a lot of in looking at the genetics and evolution of resistance, is what's called genomics. So here we can interrogate the entire genomes of our organisms to ask what mutations are responsible for resistance, as well as what mutations might contribute to lessening the cost of resistance. So we have now methods that we can use to quite easily and cheaply sequence the genomes of organisms, of microorganisms rather. So when we introduce resistance mutations into a microbe, or when we have a clinical strain that we're interested in, we can sequence its genome. Then we can figure out what mutations cause resistance, what mutations might be associated with higher or lower costs. That can help us in trying to develop, let's say, diagnostic tools, and in trying to figure out what specific mutations are relevant